So let's talk a little bit about how eukaryotic cells differ from the prokaryotic cells when it comes to transcription and the transcription process. Concept 17.3 says eukaryotic cells modify RNA after transcription. And again, think about this like a secretary or a assistant to the boss formatting and cleaning up all the instructions that the boss hastily wrote out on a piece of paper before she or he gives them over to all of the workers. So this boss's secretary may proofread his work and spell check it and format it properly, and then she may send it out in a mass email to all of the workers and give them their instructions. So that's kind of similar to the process we're going to talk about in eukaryotic cells, how they modify the original RNA strand before it gets sent out of the nucleus. Remember, in a eukaryotic cell, we have transcription and translation are kind of divided into two separate areas because transcription takes place in the nucleus and then translation outside of the nucleus. In a bacteria cell or a prokaryote that doesn't have a nucleus, this process or these two processes kind of take place in the same area and sometimes even sort of at the same time in a kind of an ongoing process that mixes the two. But in eukaryotic cells, they are definitely separated. Enzymes in the eukaryotic nucleus modify what we call the pre-mRNA, so the original mRNA before it's finalized, before the genetic messages are dispatched to the cytoplasm. During RNA processing, both ends of the primary transcript are usually altered. So we're going to find out that that strand of RNA usually has some changes made to both ends. And then also there are some alterations that are made to the interior part of the molecule. There are actually pieces that will be cut out and then other parts that will be spliced together before we send that messenger RNA on its way. First, let's briefly talk about how the ends of that messenger RNA strand get altered or modified. The 5' end receives a modified guanine nucleotide called a 5' cap. So it kind of receives, don't really know how to describe it other than I'll tell you that it helps the RNA strand get out of the nucleus and not be damaged by some of the various enzymes that are around and in the nucleus. And then the 3' end gets what we call a poly-A tail. Basically just a whole bunch of adenine nucleotides get added on to the 3' end, sometimes 50 or 100 just additional adenine nucleotides. And the function of modifying these two ends seems to be a couple different reasons. Number one, it seems to help the export of the messenger RNA into the cytoplasm, and it seems to protect the messenger RNA from some of the various enzymes that are floating around in there. If those enzymes start to kind of break down the strand, they'll probably break down a lot of the extra adenines on the end first before they actually get to the information. And then it also helps the ribosomes attach to the proper 5' end of the messenger RNA. So there is some alteration to both ends of the messenger RNA. We have a 5' cap, which is just a modified guanine nucleotide, and then numerous adenine nucleotides are just added to the end, kind of just like a buffer zone, so that if enzymes do start to work on that messenger RNA as it's traveling, they won't get to the actual prime information that we need to keep. Here's an illustration that kind of talks about some of that RNA processing here. So if we have this section right here is the actual protein coding segment. So you can see this messenger RNA is a lot longer than just the coding segment that we're going to be using to create a particular protein or a particular chain, amino acid chain. So we have the 5' cap that goes on here. Here's your start and stop signal. Here's that polyadenylation signal that we were talking about that sort of signals to the RNA enzyme that this is where you're done, and so it leaves the DNA at that point. And then we have the poly-A tail that gets added on. So out of all of this, really the only prime information is this portion between here and here. So this is the modified now messenger RNA. Still needs to have some modification in the middle, which we're going to look at, and then it'll get sent out from the nucleus. So let's talk about what's called split genes and RNA splicing. Most eukaryotic genes and their RNA transcripts, they have long non-coding stretches of nucleotides between the coding regions. So you end up having a lot of DNA and RNA that doesn't actually code for a particular protein. These non-coding regions are called introns, And they're called introns because they are intervening sequences, but they're not actually instructions for coding a protein. The other regions that are the important regions for coding proteins are called exons because they will eventually be expressed. And these are the portions that are actually read and translated into amino acid sequences. The process of RNA splicing is removing introns and then joining together exons, splicing them together to create a continuous messenger RNA molecule that is basically all good coding information for the various proteins that we want to make. So the piece of messenger RNA will have introns, that's the non-coding regions. Think of it, I guess, if you want to, as fluff or spam or filler, and then it will have exons, which are the actual coding regions that code for specific amino acids. And that's what's going to be made into the polypeptide chains to create the proteins. So in this long pre-messenger RNA piece, you have the 5' cap, you have the poly A tail, you have a large section of information right here. Some of those are introns, like this long section of pink here and the short section of pink here. These are introns. These are pieces that don't actually code for proteins. And then you have an exon here, and an exon here, and an exon here. So through RNA splicing, we're going to cut out those introns. There are certain enzymes that do this, and then weave that back together again. And so you end up with the final piece of messenger RNA is nothing but coding region plus the 5' cap and the poly A tail. Now this piece is now ready to go out to the ribosomes out in the cytoplasm to leave the nucleus. So this would be similar to the boss's message having been spell checked, cleaned up, and formatted by the secretary, and now ready to be sent out to the workers of the company. In your textbook in this chapter, they kind of go into a lot of detail about the RNA splicing, the removing of the introns and the splicing together of the extrons. We're not going to go into quite as much detail, luckily for us. Did I say extrons? Exons. So introns and exons. But luckily, we're not going to go into quite as much detail. The only thing that we want to say is that there are specific enzymes that cut out the introns and splice together the exons, and they're called spliceosomes. So they have this kind of funny name, spliceosomes, and that's a wide variety of proteins and then some other small chemical materials called nuclear ribonucleoproteins. So they're called small nuclear ribonucleoproteins. And you can guess that they've probably come up with a shortened abbreviation for that because that's a mouthful. So here's the abbreviation, which doesn't look much easier, but they have a funny little name. They're called SNRPs. So every time I hear this little nickname, SNRPs, it kind of makes me laugh. But SNRPs are these little proteins and enzymes that help with the process of cutting the messenger RNA and removing the introns, and then splicing back together the exons that actually code for the proteins. So if you ever see the word SNRP or the nickname SNRP, it's talking about these small nuclear ribonucleoproteins that help through this process of gene splicing, or I guess messenger RNA splicing, removing introns and splicing together the exons. As usual, an illustration always helps. So here we have a couple of different proteins and enzymes that kind of do this process. Here we have the original RNA transcript. It has some exon information that we need, plus some intron information that we don't. Here is a SNRP right there. It's one of the proteins that's going to help in this process. So all these proteins work together to cut this out. All together we would call this a spliceosome. They remove this portion of the messenger RNA, which is the intron. There it is now off to the side. And then they splice back together the two ends until we have our final clean, ready to go messenger RNA down here at the bottom. Good job, SNRPs. One final detail to mention about this process is we have fairly recently discovered a particular, not an enzyme, we call it a ribozyme. This is an RNA molecule that can act as an enzyme. So remember, RNA is made up of nucleic acid, and enzymes are proteins made up of amino acids. So they are separate kinds of molecules. And it was at one time thought that only enzymes could do this job that we're talking about. But fairly recently, we have discovered what we call ribozymes, which is a type of RNA that can actually self-splice RNA itself. It's like a self-splicing RNA. It can function both as RNA and as an enzyme. And so this fairly recent discovery kind of rendered obsolete the belief that all biological catalysts were proteins. At one time, it was thought that all enzymes, all catalysts for the chemical reactions of biology had to be enzymes, or proteins, I mean, and that turns out not to be the case. This also kind of indicates that RNA probably came into being long before DNA, because RNA can act as an enzyme and as a carrier of genetic information, which makes it probably, you could say, a more functional type of molecule than DNA, which really is only a carrier of genetic information. So ribosomes are a fairly new discovery, and they kind of challenge this idea that we've had for quite some time, that all biological catalysts and enzymes were made up of proteins. Turns out not to be the case. So finally, if you're like me, if we're cutting out these areas of messenger RNA called introns, my first question was, why are there introns to begin with? I mean, why does the cell go through this process of making these long strands of messenger RNA where half of it is just stuff we're going to cut out that doesn't even code for a protein? So what is the functional and evolutionary importance of these introns? And it turns out that they do have some important functions as part of this entire process. Number one, some of these introns contain sequences that regulate gene expression. In other words, they don't code for a particular protein, but they may affect the coding of a protein or how that protein is made. So they're not the actual instructions for making the protein, but they have not a direct effect, but an indirect effect perhaps on how that process gets done. Also, the fact that we have areas that can be removed and you have these introns and exons, some genes can encode for more than one kind of polypeptide, and it depends on which segments are treated as exons during splicing. So what that means is you could have a section of messenger RNA that gets cut in different places, and different areas are considered exons at different times. Well, the different ways they are cut will determine the type of polypeptide chain and protein that gets made. So one gene could potentially code for multiple proteins. This is called alternative RNA splicing. So one strand of messenger RNA could actually code for a wider variety of proteins depending on how you chop it up and splice it back together again. And having these introns and exons kind of gives you a bit more ability to have variety in that one particular messenger RNA. Finally, we've studied already how proteins are three-dimensional molecules. They twist and turn and fold back on themselves, and it's not just a long, straight strand. Once that polypeptide chain gets made, it begins to bend and twist and turn into a very particular three-dimensional shape, and the shape of the protein is very important. Well, you actually have different regions or domains of the protein. Remember, we talked about how on the protein there's a certain spot that may be an active site, or a certain spot that may be a binding site, or maybe a certain area where the chemical reactions take place, and another area that binds to the cellular membrane. Well, these are called the domains of that protein, and one may be on what we would consider the top, and one may be considered on the left or the right, or the different three-dimensional sides of that protein. In many cases, different exons code for the different domains in a single protein. So you may have different exons, part of the messenger RNA, coding for a specific area of that protein. So like, for example, here, this one whole strand is one protein, and it's twisted and bent and turned. So this is a polypeptide, or a protein right here. Now, to transcribe this, you have basically these three domains or portions of that polypeptide. Well, you have different exons that code for those parts, and those exons will be divided by introns. So we're going to cut out that intron and cut out that intron, and then that piece of information altogether will create this polypeptide chain with each domain kind of separately coded for by separate exons. So it turns out that there is actually use for these areas that don't specifically code for the proteins. They have a couple of different functions, and it's not just junk or filler or fluff like I might have described it before. Oftentimes those introns have a purpose. We're going to pause right there. So far we've talked about transcription, and now we've talked about how that messenger RNA gets modified before it leaves the nucleus. The next step is to go ahead and leave the nucleus and start the translation process. We'll take a break and come back and do that.